Hey everyone. I am going to do this pub quiz thing. Just as a little background, if you're watching this, I think I put it up on X, on Twitter, that ALS Canada at our national research forum that we do every year, the ALS Canada Research Forum. We've been doing it for many years, since 2006, 2004, depends on how you look at it. A couple of years ago, we started this thing called the Showdown, where we were asking questions, trivia questions. We have buzzers. It's kind of fun. We originally started asking the clinicians basic research questions and basic researchers clinical questions to see who was the ultimate winner. And the two finalists, Dr. Christine Vandevelde from University of Montreal and Dr. Geneviève Matt, the clinician at CR CHUM, or the CHUM, University of Montreal, were the finalists. Geneviève won, but then they teamed up to defend their title, to try to win the title last year, against Gary Armstrong, who got kind of sucked into it last minute, and Vincent Piché-Martel. They crushed them last year. And then this year, we actually had a team with a postdoc and a student, Alicia Dubinsky and Esmita Ghosh, and they faced off against Janice Robertson and Michael Strong in the semifinals. Esmita and Alicia won, and then faced off against Geneviève and Christine, and Geneviève and Christine won and defended their title. And we have this golden pipetoscope trophy that was basically a stethoscope I bought on Amazon as cheap as I could, and then and a pipette, the same thing, and I just glued them together in this weird way and spray painted them gold. Anyways, it was fun. And it was a way really designed to bring people into the room early at the research forum, because traditionally we have a networking evening on a Sunday night, and then on the Monday morning we have talks that used to start at 8:30, and it would be half empty room. It just wasn't fair to those people, so we kind of did this ruse where now we started it at 9. We give it the poster awards, etc. And then we were like, if we do this showdown thing where we're getting everyone hyped up to come and watch the sort of game show with the winners getting this silly trophy, that it would bring everyone to the room. And then when we started the actual talks at 9 o'clock, it'd be a full room. It worked really well that way. Anyways, so this year, before the thing started, I'm the one that asks the questions. I ended up just putting together, I don't know, 60, 70 questions, whatever it was, writing them down, and you sit down in a word and say, well, what's a good question? I'll ask about, C972, something to do with that. And then typed out the questions, and then printed them off, and those are the questions that got read. And then, just to be fair, because I don't know how biased I was when I actually wrote these based on my train of thinking, they went through a randomizer. So that's why what you'll see if you saw the sheet is that there's different numbers on them. That was basically the numbers in which I just typed them out, and I came up with them, and then I hit the randomizer, and that's the order they came up in. That's also because, if there's questions about older things, you don't want the student and the postdoc to get those, and then Mike Strong and Janice are fed all these questions that they know because they're a little longer in the tooth, and you also don't want clinical questions only to go to the clinicians, etc. So all that said, if you're here, you might have actually watched or tried to go through those questions. And so anyways, these questions were done. They were written. We got through a certain number of them in the timer that we had on the screen. And then I realized I have all these questions and I don't know what to do with them. So I thought it'd be kind of fun to just do a pub quiz style. I don't know if anyone's going to watch this, but I don't really care. I mean, it's not a big deal. I'm just going to put this up on YouTube and link to it. And if anyone finds it interesting, they can watch it and see how well they do. I think, again, this shows you how much effort I've put into this for this particular pub quiz thing. I think when you add up all the questions, it's supposed to be 84 points. I don't know if that's exactly true. If somebody's getting 82 and they're worried that, it was actually 82 out of 82, then good for you. And also, how do we get you a job in the field somehow? Or maybe you already have one, because that would be incredible. Anyways, I was also told by one of the contestants, I'm going to share my screen here because we're going to just put it up, that they were really hard this year, the questions. So That's why I also thought it'd be kind of fun. You know what? I don't see the share screen function. Oh, there it is. It's right there. Sorry. Okay. Yeah, I was told that it was kind of hard this year. So, I guess we'll see how hard it is. We're going to go through them and we'll just give the answers. And in some cases, I guess I know a little bit more about the things. In some cases, just a trivia question. So, all right, let's get going. This was round one. This was one of the questions that ended up coming up for the semifinals, but we're just going to treat this as one long pub quiz. So the Showdown 2024 pub quiz. You ready? Here we go. You can check your answers based on this. I'm sure you've been able to look them up on the internet if you did this, or if there's anyone ever watching this. That's also possible that no one's ever going to see this. So, okay. So first of all, what was the name of the compound that reached clinical trial for reducing levels of SOD1 led by Dale Lange before the ASO gained momentum? The answer is pyrimethamine. This was work that Dale actually did with the Swedish individuals, Peter Anderson, probably Stefan Markland. I'm trying to remember. The Swedish know SOD1 better than anyone. Peter Anderson probably knows more about SOD1 than almost anyone. So this thing actually reached a phase 1 clinical trial. They were looking in I believe CSF for plasma. I'm trying to remember. Anyways, it lowered. There's probably a paper back in 2015 or something. It lowered the levels of SOD1. So that's kind of cool because in the end, that's what tofersen does, and what other new things that are coming along, such as Unicure's AAV and Regeneron's siRNA, all designed to lower SOD1. So this was a sort of first kick at the can. I'm not sure what really happened with it. It's a good question. Should ask him next time I see him. But anyways, pyrimethamine is the answer for number one. Shoot, there was something Bob Brown did with pyrimethamine too. I think he struggled to get it to work or something like that. I don't remember. You can look it up. Bob Brown, pyrimethamine. There's something there. I don't remember. Can't know everything. Okay, next. A knock-in mouse of what gene that has variants conferring ALS demonstrating a motor unit defect phenotype is currently being pursued by Steve Kolb at Ohio State? It's KIF5A. So he has essentially made the knock-in mice with the variant, the mutation, and has seen this sort of motor phenotype. I think there's splicing phenotype that's in there too. And so, for anyone out there, what does that mean, a knock-in mouse? So basically, when we want to make a model of ALS in the lab, and we make these mice, for many, many years, we made what are called transgenic mice. And basically, you're adding a piece of a gene, or adding a specific thing to the genetics or to the different cell types in the mouse. And that makes a whole lot of that particular protein in the end that you're trying to make. So, for example, in an SOD1 mouse that we use, typically the most common one's called G93A. This will make a really high level of SOD1 protein. It's just the way that these transgenic mice work. They sort of overexpress a protein more than the functional level. But as we've developed what are called gene editing techniques, and I'm sure many of you have heard of CRISPR-Cas9, we can CRISPR or gene edit in changes to the genetics actually in the gene of the mouse, so that the level of the expression of, or the level or the amount of the protein that's made is actually what we call physiological. It's the amount that should be there, but it has that genetic change. So you can actually see what happens to the mouse if it were akin to what's happening in humans a little better than if you're just blasting away this protein. Because when you do that, you don't necessarily know if the problem is that you're making too much of this protein and it's throwing everything out of whack. KIF5A knock-in mice, Steve Kolb's seen, what we call a phenotype. So it's not necessarily getting classically ALS as we see it, as I understand it, but it's getting some symptoms or some this phenotype that kind of mimics certain things in ALS that could, it helps to tell us. It's another one of those tools to say, okay, we know that KIF5A variants cause ALS in people, or can cause ALS in people. And so if we see what happens in mice with them, and then we see what happens with, other things like C9orf72 and TDP43 mice and all of this, what can we see that are the sort of correlations between them that might be avenues of where things are going wrong and that we can actually put treatments against? So anyways, obviously I could just keep going on that, and we probably don't want me to. So we want to get to question 3. Elatam, Lainals, and Unella are consortia advancing ALS in what continent? South America. Yeah, Elatam's really cool. This is a new network that is really to try to bring clinical research, clinical infrastructure, clinical trials to South America. Martha Pena, who's on the International Alliance's Scientific Advisory Council, she's awesome, out of Bogota, is sort of leading this. It's had, support over the years as well in sort of building some infrastructure in South America, from Monica Povedano in Barcelona, and Orla Hardiman in Dublin, who have also been working on Lainals, which is sort of an epidemiological study that really started the idea of getting some clinical research happening in South America. So that's really cool. And Unella actually super cool too, because that's all the organizations from South America kind of coming together in a unified consortia, which is really important and powerful, especially in these underrepresented, underserviced areas, to be able to have that, to share best practices, stay on top of what each other are doing, help build each other up. I think South America is just going to be a really blossoming place for ALS in the years ahead. Hopefully we start to see more trials get there, more clinical research happening, more focus on people in South America who are living with ALS and want access to research, and then obviously part of that is building in capacity to market therapies in new ways, so that when things are great and coming to places, we want to see those happen around the world. And we'll talk about other places, I think, in some of these other questions around the world. So yeah, South America. Okay. I'm probably going way too slow. That's fine. This is my pub quiz. I don't think anyone's watching anyways. What was the treatment given in the clinical trial, quotations, that Lou Gehrig participated in? The answer is vitamin E. So Israel Weschler's a physician in the 1940s who had vitamin E trials. Vitamins were kind of new. The reason why I put clinical trials in quotations is because I think the first sort of formalized clinical trials were happening with tuberculosis in the late 1940s, where they sort of had people that were in one room. It was sort of controlled for. It was like the first sort of controlled clinical trial. In this case, they were just giving it to people and monitoring them, and Lou Gehrig was part of this. I think even you can find some documentation that Lou Gehrig was saying that, maybe it improved certain things. There's actually some interesting media stuff from back then. I think there's a New York Times article where Dr. Weschler is talking about vitamin E being like helping, almost curing 2 people or something. So, hype was a big thing even back then. But obviously vitamin E is not something that is a disease-modifying treatment for ALS. But really, an interesting thing that this was something that actually, let me say this. I think what's cool about this, or what was really monumental or important about this, is that if you read some of the literature as well, and I may be wrong about this, someone can correct me if they want on X. No one ever does that. It corrects me on X. But yes, you can absolutely, please let me know if I'm wrong on this. But when Lou Gehrig was diagnosed, this actually really led to the idea of needing to do research on ALS. This stimulated the idea that we need to start giving people things to try to treat this more in a more organized fashion and with groups of individuals, because before that it was, kind of more obscurely known to neurologists, but it wasn't something that, spearheaded this movement to learn about a disease and sort of start to do it. It wasn't really dedicated ALS researchers until like the 1970s. The evolutions that happened in the 50s and 60s really came from, I think, spearheading this in the early 1940s with Lou Gehrig. Obviously, World War II was happening, so it was a tough time and really rudimentary what you can do in research. Okay, next. What is the name of the neurons that primates have and have postulated in humans that Andy Eisen, so some of this Canadian focus, by the way, awesome Canadian researcher legend Andy Eisen suggested could be key to understanding dysfunction in ALS? The answer is mirror neurons. So mirror neurons are something that have been shown in primates, in monkeys. I don't think they've been confirmed in humans, although I haven't looked at literature whether someone's done this in recent years. But the idea was that some of the things that happen with like cortical motor neurons, cortical spinal motor neurons, could be mimicking what's happening with mirror neurons. It's interesting stuff. He's got some really cool postulating papers that are out there, a lot with Martin Turner, Matthew Kiernan, Roger Lemon. Worth having a look. He's a really fascinating guy. Actually, really should get the credit too for the fact that he's really one of the founding individuals for the idea of upper motor neuron based dying forward hyperexcitability in ALS. Really doing, I think TMS and stuff like that in the 80s on ALS and just maybe it was the early 90s. One of the first people ALS Canada ever funded for research anyway. Lots about him. Big leader out there for BC ALS, and I think one of the first multidisciplinary care clinics, the second one in Canada behind Arthur Hudson in London, and one of the first ever, probably 1980, 1979. If you talk to, Terry Hammond Patterson, Stan Appel, this was really, early, very early, around their time, earlier than them, maybe even multidisciplinary care in Canada. Big leader. Arthur Hudson, 1977. Also probably the second one ever, second or third, maybe Forbes Norris and Ted Munsat at California and Tufts. Okay. What is the name of the famous rugby player for the Leeds Rhinos, currently living with MD, who has spearheaded massive awareness and millions of pounds raised for research? I should say that this was written, and I'm just reading this now. Again, this was written in April. The answer obviously is Rob Burrow. I'm sure many people have heard. So not currently living with MND. In fact, he passed away very recently. Just incredible advocate for MND, someone who obviously, was in the spotlight in the UK for being this incredibly tough, small guy, just like a tank, and then to very publicly show what MND did, how his progression with the disease was very publicly with his family and stuff, obviously was really powerful for a lot of people, raised a ton of money, and also highlight, go watch the documentary on this. Heartbreaking. And then his friend Kevin Sinfield, if you haven't heard about the ultra marathons, I think he's doing another one soon. It's incredible, raising tons of money. He's like a real life Marvel superhero, that guy, doing things to raise money. So the answer, Rob Burrow. Okay. Next. What is the name of the ALS gene that operates in the same pathway as NEC1 and, like C9orf72, only has a designation by its chromosome and open reading frame numbers? C21ORF2. So this was another one that was sort of first described by Project Mine. I think it's in DNA damage repair. I don't fully remember. Yeah, something like that. But not something I think is even in like the ClinVar, ClinGen databases yet. So I don't know how much is being done on it, but I think there's some work happening here and there with C21ORF2. Anyways, just thought it was interesting. I think actually, like I think it's also got something to do with the eyes. A lot of the ALS genes actually have something to do with the eyes, optoneurin and things like this. So that's interesting. I could be wrong on that. And if you look it up, I could be wrong because, again, I'm just sitting here trying to riff on this. Okay, next. What is the name of the MLB analyst currently living with ALS who uses the tagline, "Baseball is the best"? Perfect timing right after Lou Gehrig Day that I'm doing this. Sarah Langs. What a joy it is to see her in my X feed, doing all these cool things with baseball. And I love these things where it's just like a game turns around at the end of the day, or at the end of the game, completely, someone ends up winning at the last minute. And she's got this sort of like roller coaster of the way that the odds were going for the winning team and stuff. And just seems like an incredibly wonderful person, and huge ambassador for ALS in the United States. So that's Sarah Langs. Gotta buy one of those t-shirts. Cool t-shirts, I think, this year. Baseball is the best. It is the best. I love baseball. Okay. I'm wearing a hat. This is not a Boston Red Sox hat. I got asked this at an ALS walk this weekend. It's Buffalo Bisons, which is AAA team for the Blue Jays. I'm a Blue Jays fan. Sorry to anyone who's seeing this who doesn't like Canada. They're not doing great this year, but like this Bison's hat. There's Buffalo. There we go. It's not all Canada. Okay. Name one of the following. Actually, you're gonna have to name them all because it's a pub quiz, right? So this was for the buzzers and all that stuff. The hot sauce, the ALS Ice Bucket Challenge lineage can first be tracked back to the person living with ALS to be connected to it first, the first person living with ALS to be connected to it, or the Canadian former NHL Bruin who first stimulated it from grassroots to viral. First answer is Carmelita's hot sauce. I don't remember. I think they're in California or something. But the actual ALS lineage, if you go back, it stops there. Unless you are Daniel Valencia and you know who challenged who or where it all started, which we can never figure out. Yeah, that's the farthest you can track it back. That was actually somewhere in, I think, June. So it obviously tracked back further, or else they started their own, and that was the end, the way that it ended up. It wasn't the PGA Tour one. It wasn't the one that went to the Today Show. It was actually this own lineage that went through long drive competition individuals. There was still some golf, but it wasn't like the famous golfers. And that leads us to the second answer, because it eventually went to Chris Kennedy, who challenged Jeanette Senerchia, his cousin, I think, and whose husband was Anthony Senerchia. That is the first person with ALS from that group that led to Pat Quinn for the win, and Pete Frady's Team Freight Train, and that led it to go to the last answer, which is Greg Campbell. Greg Campbell from the Boston Bruins was friends with Joe Correa, who was also friends with Pete Frady's, and I believe there's something to do with a public address announcer or something like that. But anyways, got convinced that he should do this. He ended up challenging, I think, Milan Lucic, or was it, it might have been Marchand. I don't remember. It's in the lores of Google. You can look it up. Anyway, started challenging other Bruins. Bruins started challenging other people. The Patriots did it. It went around Boston sports. It ended up going viral, and this is really the trigger. It went from grassroots with the, Pete Frady's, Pat Quinn, Anthony Senerchia in Boston and New York areas, and then once it was Greg Campbell doing it, I think it was his Muskoka Cottage, so Canada, yay, challenging Bruins. That's what led it to go viral, really, because it was like a couple of days after that, each of the Bruins did it. They challenged other people, and it just went crazy. So super cool story, actually, how it all went. But you've probably all heard it. You don't need me to say it again. Okay. I don't know what number on. It's 31. So I already lost track because I'm just rambling. What two proteins have ALS causing loss of function variants and are intimately linked as part of the innate immune response and autophagy? Optineurin and TBK1. Lots you could talk about with both of those. I'm probably not going to go into it too much. You can look them up. I think TBK1's probably that like fifth member. If you talk about what most of the ALS research is done on, it's C9orf72, SOD1, TDP43, and FUS. TBK1's probably the one, maybe NEC1, that must be missing something, but like in terms of more of the like definitive ALS genes that are being worked on with different models and trying to understand what's happening. It was actually a TBK1 model, I think a loss of function TBK1 model that led to the idea that RIPK1 was activated, which led to the Sanofi. Well, Denali, I think, had a RIPK1 inhibitor that Sanofi then put into the Himalaya trial and was recently negative. So, but that's very downstream. That doesn't mean that we can't learn a lot from TBK1. Anyways, yeah, I'm rambling again. So we're going to go to question 23 because I don't know what number one. What is the currently used terminology by Klinjan Kimvar for the common SOD1 variant in North America that confers rapid progression? Quite commonly, people used to call it A4V, and now I think you're really supposed to say A5V. I think it must be that we didn't count the original methionine So it was A4V. But A5V or A5T could work because that's also very rapid progressing and does exist. I think A5V is the one that kind of gets most often talked about. Yeah, rapid progression, and something that we hope the ATLAS clinical trial is gonna make a big difference for. If somebody has a neurofilament spike, they've got something like an A4V variant. Could you imagine if you could start it early enough to actually stop them from ever getting the disease or slow it down massively with such a rapid progression? It'd be incredible. Probably the thing I'm most excited about in ALS right now, because it tells us that, right time, right treatment, right target, we might be able to do incredible, incredible things. Okay, number 56 at this forum, we had one of the first women leaders in multidisciplinary care for ALS. Oh yeah, Orla Hardiman was the opening speaker talking about precision ALS, Professor Orla Hardiman from Trinity College Dublin. It was awesome. Who is the first woman still practicing today to begin a multidisciplinary care or ALS specialist clinic? The answer is Terry Hyman Patterson, who was at Hanneman College at the time. She's been to Drexel, she's been to Jefferson, and now she's at Temple, all in the Philadelphia area. She's awesome. You're not going to find a neurologist who cares more about her patients. And I think she said she'll never retire because she just cares too much about her patients. She also done a lot of really great work in the lab in terms of trying to understand what the importance of the backgrounds on mouse models is in how we may be misinterpreting mouse models. So in terms of like what strain of mouse that it's on, because it actually matters, and also differences in male versus female mice and stuff. She's done some really cool stuff in the lab, and a clinical trialist, just an amazing person. I used to sit next to her at a lot of conferences and ask her opinions on things when I was really new in this role. And I really appreciated that she always let me sit next to her. She's like, oh, you again, question guy. Great. Number 20. What percentage of NFL decrease is the threshold for a positive response being used in the experts ALS platform to overcome assay variability and provide assurance of decrease? Value is 30%. So, I mean, depending on who you ask, I think that there's about a 20% variability in the assay. So you need to have higher than that, or else you can't tell for sure if it's error. And you want something that's robust enough. Experts is this really cool platform. You should look into it, where they're looking at neurofilament decrease as sort of the major readout to build a pivotal trial around. So they're not saying that this definitely says that it works, but it's like looking for those home runs, those things that are preserving neuronal integrity, that could then be built around a trial that looks at function and all of the different things that we need to look at in a longer trial, but trying to really, stack the deck for something that looks very promising, as opposed to traditional phase twos where you have very ambiguous data moving into phase three, and hopefully we're sciencing those a little better to be able to get better understanding of that. But experts ALS, super cool. Chris McDermott and from Citran South in Sheffield and Oxford, Martin Turner, really spearheading that, part of the UK M&D Research Institute. Also, if you don't know about that, look that up. Super cool. It's also a big movement of collaboration between organizations, academics, and the incredible community of people diagnosed with M&D and their loved ones in the UK. Patients United to End M&D, but it's not patients, right? It's United to End M&D. I'm forgetting what else is. David Setters, Lee Millard, awesome people, super cool. Nicola, all incredible people. Rob Burrow's dad, just more. I'm missing a lot of the people who are awesome. Going over there soon. Looking forward to seeing a lot of people, hopefully, at Encourage event. Look up the Encourage event. Super cool for young ECR researchers. Okay, I'm babbling again. 54. Which famous golfer's longtime caddy was diagnosed with ALS in 2003? That's Tom Watson. Bruce Edwards, his caddy, longtime caddy, was diagnosed. There was a huge poignant moment at the U.S. Open one year. I don't remember what year. It might have been 2002 or 2003. When he had already been diagnosed. And I think Tom Watson, being really kind of older at that point, being on the senior tour, was like leading the US Open. It was a big deal. Really cool. Raised a lot of awareness. I think Tom Watson still does a lot of things for ALS in the United States. Super cool. Okay, number 7, in a 2020 manuscript looking at real-world evidence of riluzole, Eric Pioro, now in Canada at UBC, Jinsey Andrews and colleagues from Niels found that it was more effective than we perhaps give it credit for. Median survival between riluzole and riluzole-free individuals ranged from 6 to how many months? The answer is 19 months. 19 months is not insignificant. One of the things I've tried to ask people about was whether or not you could look into who are those people that are getting 16, 17, 18, 19 months. And then can we figure out either A, whether they need to definitely be on riluzole, or B, are they slower progressors perhaps? And maybe, some of these drugs that are not necessarily targeting a top pathway may be more effective in slower progressing, and those are the individuals who are never going to get access to them because we run clinical trials on faster progressing people who need to see the change in a clinical trial, and then when it goes to market, like with the Darvone or with AMX35, when it came to market, it was pretty much the clinical trial group that got access. But what if the disease, what if it was similar mechanisms in slower progressing individuals and they stood to benefit even more? If we look at tofersen, I mean, it's not a far of a stretch. Individuals who tend to be at the slower progressing end of things or more moderate progressing seem to be having stabilization and some regain of function. So that's something to look into. I think there's a lot to learn from riluzole. Also, what it's doing. We've always surmised that it's excitotoxic blocking, glutamate affecting, I believe. But I should know. My gosh. Anyways, it's a lot of stuff just to talk about here. So anyways, what is it really doing? How is it having this time-tested effect through real-world evidence? It's a good question. Okay, number 57. There were 2 well-known geographical areas where ALS Parkinsonism dementia complex, PDC, was known to be endemic. One of them is the island of Guam, and the other was what peninsula in Japan? The Kii Peninsula, KII. Oh, the Guam story is really interesting. And there's actually a gentleman from Toronto, from Canada, who did a lot of work out there, moved out there, recently passed away. I'm blanking on his name. That stinks. I feel terrible about that. You should look it up. It's a really cool story. The Chamorro tribe, I mean, as I understood, they were pushed into the mountains by the war. They ended up eating a lot of cycad seeds. There's a hypothesis that maybe it was the powder that they got from the cycad seeds was actually causing this sort of ALS PDC. And then also, one of the ceremonial dinners was a stewed fruit bat in milk, coconut milk, I think, or something. And the fruit bats would eat the cycad seeds as well, and that would bioaccumulate in their tissues. And this was sort of an endemic thing that happened in Guam, and I think it's been mostly eradicated through dietary change. But that's a good question. I got to look all this up. It's really interesting. I looked at it like 10 years ago a lot, and then I think when I realized that this wasn't necessarily going to, maybe it does, tell us about how we're going to fix more classical ALS that we're looking into in a pathological sense now. Probably haven't looked into it enough. Anyways, lots to look in there too. Okay, number 44. In 2014, a group led by Chris Henderson implicated low levels of what protein to confer resistance in oculomotor and Onof's neurons using gene expression data from SOD1 mice. 2 years later, Eva Hedlund, Sweden, implicated higher levels and activity of what growth factor conferred lower vulnerability in resistant neuronal subtypes of SOD1 mice. So with Chris Henderson's, it was MMP9. They found that when you had a lot of MMP9, it wasn't that's what you found in the vulnerable neurons, and that lower levels in the oculomotor and Onof's neurons, which are more resistant to ALS. That's why eye gaze is a big thing in ALS, because the oculomotor neurons are more survived. Your bladder control is still there with ALS because of the Onof's neurons, and they had low levels of MMP9. And so it was hoped that that might be an actual therapeutic target. I remember in 14, 15, 16, somewhere in there, speaking to Bob Brown at a Foundation Andre Delhomme conference about MMP9, I think, and asked whether or not that had been moving forward. And he said, I think that they were struggling to find that if they knocked it down, it wasn't having a massive effect in the animals, so they weren't sure. But it's interesting to see. It'd be interesting to see how it works in like an iPS-derived human cell now. It probably has already been done, and I'm not thinking of it. I do know a few people. Sylvia Pozzi in Canada is looking at MP9 in certain ways. Still, maybe an interesting target to reevaluate, unless there's something in the literature I'm completely missing, which is entirely possible. Can't know all the literature. Ava Hedlund then did, I believe, iPS-derived oculomotor neurons and saw that they had high levels of IGF-2. And that might have been another thing. So if we could boost IGF-2, is that something? It's very generic in the sense that I think we've looked at IGF-1 and other growth factors. I think IGF-1 was a big Fred Gage paper, Rusty Gage paper back in the day, where they injected muscle IGF-1 or something, and it would have protection. Anyways, IGF-2, that's your answer. I'll go to the next one. Number one question was in the this is obviously the first thing that came to mind when I wrote these, which is a little crazy. In the mid-2010s, a clinical trial of what monoclonal antibody treatment was run in ALS by GSK, GlaxoSmithKline, but ended up as a no-go. The answer is ozanezumab, or as a lot of people pronounced it at that time, ozanezumab, or ozazazazaz, and then they just kind of kept talking because no one wanted to say the name ozanezumab. This targeted the no-go-A receptor, and that was essentially that pathway was what's expressed, I think, in our neurons when we first are born and we're developing, and our neurons kind of stretch out to touch our muscles. Once they touch our muscles, you don't want them to keep growing into the muscle. They've made that sort of neuromuscular junction. So there's this expression of no-go-A, and to sort of stop it. So the idea was that if you could inhibit that pathway, could you make the neuron kind of get that regrowth bit to it? It didn't work. Interestingly, actually very topical, we know that very recently Biogen and Ionis stopped the ataxin-2 clinical development after negative data from a phase 1. And it was Aaron Gittler had a paper where it was the no-go-A RTM4 pathway when inhibited reduced ataxin-2 levels. So it'd be really interesting to see if ozanezumab from that clinical trial actually reduced ataxin-2, and maybe we knew ahead of time that reducing ataxin-2 wasn't going to work. Obviously, we don't have the data from it yet, so maybe there's still something there for ataxin-2. We don't know how much it was knocked down. We don't know all the details, but if Biogen and Ionis were willing to stop it, that's certainly a big standpoint to say that maybe ataxin-2 isn't the way to go. We don't know. Time will tell. Early days. Also, big thing to say in there about gain of function models, the TAR44 mice where ataxin-2 decrease affected 3 different mice in a good way, and then it didn't work in people, perhaps. So lots to talk about there. Not now. Question 14. A former regular attendee to this meeting, that's the ALS Canada Research Forum, and collaborator of Charles Krieger. What is the name of the researcher and technique used in exploring how shared vascular systems can help us understand immune and inflammatory aspects in ALS? The answer is Fabio Rossi. Really brilliant guy, but he does this parabiosis thing where you sew the vascular system of 2 mice together, or 2 rodents together, I guess probably any animals, and then you can see whether or not a change in the circulatory system in one animal affects the other one, which it's very interesting scientifically, right? So the idea was rudimentarily, if you put an ALS mouse and you parabiosis with a non-ALS mouse, could you see if there's transmission of the disease through the bloodstream from the mouse that has it, for example? But more in the immune inflammatory respect, it's that if Tregs, for example, were boosted in one, would that actually show that it's through the circulatory system that something's happening, because it would be shared to the other mouse, and can it affect whatever's happening in the other mouse? So it's just a way to be able to really look at whether or not the immune inflammatory, a different way of looking at whether the immune inflammatory systems are responsible for certain things happening in biology. And I'll stop talking on that because I'm clearly not an expert on parabiosis. Okay. Which Canadian researcher was praised for his foundational work by both Clive Svensson and Kevin Egan in a virtual discussion panel run by ALS Canada during the pandemic? Well, it's on YouTube if you want to go see it. Good shout out for during the pandemic when we did a Biogen-sponsored series where I just had the best time because I got to have these 90-minute conversations about different topics, animal models, iPS cells, clinical trials, therapeutic targets with just incredible people, and the sort of modeling human ALS in the lab one was Rita Sattler, Kevin Egan, Clive Svensson, and the answer to this question, Victor Rafuse, Vic Rafuse from Dalhousie in Nova Scotia, who really was one of the forefathers of the individuals who first put together work on developing human motor neurons and what was needed to make motor neurons in the lab in a dish. And he got a lot of praise for that from legends like also big people in the field, Clive and Kevin. And that was great to see. Because I know there was work done in Colombia a lot as well on this at the time, but I know that that Vic was a huge part of that and something that they all found very important work to theirs. So that was super cool. Go watch it on YouTube and watch the rest of them. They're older, but they're super cool. Okay, 27. We're now into what I had questions for the final. We had double the amount of time, so I just figured we would have more questions. Sounds like there's a lot. So let's just go through them. No one has to watch this, so you can stop and come back. See how you're doing. How are you doing, by the way? It's 25, I think, is the points to this point. You're all 25, right? Okay. Worked on by Xiong Sun, Sammy Barmada, and Patrick Dion in ALS. What is the name of the most abundant epitranscriptomic signature? What's epitranscriptomics? Well, we actually talk about proteins getting modified by things like phosphorylation, sumoylation, or all kinds of different, what we call post-translational modifications, ubiquitination, and RNA also gets modified, making everything that much more complicated. And it's actually something we haven't spent a ton of time understanding and working on in biology and in ALS, but these three individuals have done that. And the answer is M6A, which is N6-methyltransferases, methyladenosines, N6-methyladenosines. There's things called writers, erasers, and readers, different enzymes or whatever those are the methyltransferases, the demethylases that are part of like adding and removing these. Kind of like a kinase phosphorylates and a phosphatase gets rid of the phosphorylation on a protein. In this case, you've got writers and erasers in an RNA. Just much more complicated. Anyways, a great paper from Xiong Sun not that long ago. Patrick Dion had this idea that epitranscriptomics were involved in ALS a long time ago. I'm going to say 2015 or 2016, maybe like something like that. So him and Jay Ross, who's now in Aaron Gettler's lab, did some work on that. And Sammy Barmada has a nice paper on M6A and ALS as well. Sammy's awesome. Yep. That's the answer there. Number 50. Name one drug tested in the Healy platform. Name one drug tested in the M&D Smart clinical trial. Oh boy. Okay. So this could be anything. So we're going to have points for anyone. If anyone gets, there was Zilucoplan, which was Raw Pharmaceuticals, became UCB. It's the complement C5 inhibitor. Didn't work out. CNMAU8, still in development with clean nanomedicine. That's the gold nanoparticles. That's supposed to boost the bioenergetics of the cells and a few other things. Looking to get that into a phase 3 clinical trial. They also had the Australian trial on that with Muni as a readout. Muni. Matthew Kiernan's a big supporter of the CNMAU8 program. I think he's the PI in the phase 3. Okay, there's that. Verdiperstat also was negative. Myoperoxidase inhibitor. Why am I blanking on who did the verdiperstat? People are yelling at me probably if they are watching this. Shoot, I know the company. I'm trying to think in my brain. Anyways, doesn't matter. Points aren't given for the company. Pridobidine, which is Prelenia, which is also still in development after their Healy trial readout. This is a sigma-1 receptor agonist, which is shown in a lot of mouse models to have value in ALS and other diseases. neurodegenerative diseases. Yeah, and so the jury's still out. I think they're planning to run a phase 3 clinical trial to see. There was some speech benefits, I think. It was hard to tell. It's phase 2. It's small. These are 6 months, phase 2. It's hard to really tell what's going on with it. So I think that's their plan moving forward. Okay, so I've said zilucoplan. Shoot, I've said verdiperstat, CNM-Au8, pridopidine. There's trehalose. Obviously, was supposed to be an autophagy activator. It's been around for many years like that, and that was Celose. Hopefully we try other things in autophagy that this doesn't kill it because it was also negative. ABBV, CLS 7262, and DNL 343 from Calico and Denali, respectively, which are EIF2B agonists. So they're blockers of the ER stress pathway. Which is interesting. It's a counter to another one that's happening elsewhere that's activating the ER stress pathway. So we should get answers on both of these, hopefully with good target engagement. We'll be able to look at that. So that's the Healy platform trial. I think I remembered all of them, but whatever. That's not bad. It's more than one. And then the M&D Smart platform, which is in Scotland. There was memantine, which is an NMDA receptor antagonist. There was trazodone, which I think prevents the ejection of serotonin. Trazodone, actually, Aaron Heusler did show something to do with DPRs, although now that's a whole other thing I won't get into because we know that'll be a big tangent. I think so. For C9 with trazodone. Anyways, trazodone, I think both of those were negative in M&D Smart. I can't remember. Memantine was actually tried in Alberta in a clinical trial that we funded. Didn't pan out. It didn't get published, which is a problem because then I don't know if that got published, whether it would be known when they ran it in M&D Smart. And now it's amantadine, which is a anti-dyskinesia or something. It's a dopamine agonist, and it does a bunch of other things. And that one actually is interesting. I think that was in a clinical trial by Ted Munsat, who was one of the first people to start a multidisciplinary care clinic, like in the early 1980s at Tufts, and it was negative. But maybe it was felt when they put it in M&D Smart that it wasn't tested properly or something like that. But so amantadine is the one that I think is active now. The M&D Smart's an interesting one. It's a wider eligibility criteria. It was really set up to be very participant friendly. And the two active platform trials, the Magnet platform trial with TriCals is a little confusing how it's a platform yet, but I know that they're developing things that are going to be really interesting in terms of trial design and platform type things in the future. With all the incredible stuff that's happening over there with TriCals and the science that's happening. Again, big tangent if I go into that. So number 58, what type of programs are the vast majority of ACT for ALS funds designed to support? A lot of people know this. It's expanded access programs. EAP for things that are at the doorstep or in phase 3. Lots to say there. Anyways, I think at the end of the day, what's amazing about that is, it perhaps we don't have the best things that were in the phase 3 realm at the time to necessarily warrant that. Obviously, it's important that people feel like they have access and have a chance. I think scientifically, the concern had always been that perhaps we just didn't have things that we were really excited about. Companies were excited about, maybe we weren't super excited about them, that were getting into that sort of realm where it would be eligible. But that has been something that is, currently moving forward. So people are getting access to experimental therapeutics to it. But let's also say that some of that stuff's been retooled into things like AMP-ALS and all ALS. And that's incredible because, those are also big deals that are going to make a big difference because the huge amount of funds that were achieved by the massive outpouring of collaborative effort by the community to make this happen. And certainly IMALS is a big part of that, big push on that. To get that through is paying off in other ways as well. So that's a huge credit to that for sure. Okay, who is the Baseball Hall of Famer that had ALS is not Lou Gehrig? The answer is Catfish Hunter. He was really good in the early 70s. Oakland A's and the Yankees. I think he had like 5 straight seasons of 20 plus wins. So for a period of time, like incredible. And I think he won a bunch of World Series when the Oakland was like unstoppable in the early 70s. So Catfish Hunter, not always recognized as much as probably he should be, although I'm sure there's got to be some local society somewhere that's like named after Catfish Hunter and something like that. I'm sure. And I'm just not aware of it right now. Maybe I've seen it in the past, but I can't remember exactly. And I'm sure they're not watching and yelling at me that I should know, but if they are, then I'm sorry. Okay, number 28. What well-known PI was the first author on a publication in 1994 that indicated SOD1 variants were likely a gain of function? The answer is David Borchardt. I think this was in Don Cleveland's lab, right? So I guess that's an answer that could work. I don't remember who the middle authors, maybe Jeff Rothstein was in there somewhere. Probably was. But David Borchardt, I believe, is the first author. He ended up becoming an incredible PI in Florida, where he works on, understanding better how SOD1 propagates, how it works biologically in our systems, really pushing that idea of gain of function. And that's critically important, because the thing is discovering that this was a gain of function and not loss of function is why we have something like tofersen, because knocking it down, try to relieve the pressure from that gain of function. Yeah, there's a whole lot to unpack there. People, you can talk about loss of function, etc. But yeah, I mean, in terms of the original sort of paper that highlighted that, when I think it was when you knock it out and you cross the knockout of SOD1, or you just have a knockout mouse of SOD1, doesn't make SOD1. They had some sort of illness, but it wasn't the same as when you had the variant or the mutation or overexpressed or whatever. So it just wasn't what they felt was a model of ALS. So it wasn't felt that there was a loss of function. But again, lots that we could talk about there that I won't, because I'll move on to number 30, which is that Stan Appel once had a review article on, so Dr. Stanley Appel from Methodist in Houston once had a review article on calcium where it was described as what fictional villain with relation to ALS is Darth Vader. Not much to say there. Darth Vader's, it's a villain. Calcium's a villain to our neurons when it's improperly balanced. So yeah, look it up. PubMed. Darth Vader, Stan Appel. Comes up with a result, probably. Should. Number 41, a recent study led by Carolyn Young in the UK suggests that ALS-FRSR should only be used as an outcome measure following transformation to an interval level. Following transformation to interval level data, and treatment should only be considered effective after this, with what point difference from the placebo? The answer is 5. 5-point difference when transferred to the interval level. And this would be an appropriate way to use ALS-FRSR. Carolyn Young is awesome. She probably deserves even more visibility in the field. And, one of those people that is going to be important as we move forward in trying to better understand outcome measures and how we're going to work with the ALS-FRSR. I know it's a very common thing to just say down with the ALS-FRSR. I mean, it's not probably going to go completely away, but what is it? What are we going to do? How are we going to use it? How are we most effectively going to use it to get the value out of it while trying to find other outcome measures for function in ALS? And this is a very recent paper, a really cool one. One of those ones alongside, others like Ruben Van Eck and very many others who are looking at the better way that we can use ALS-FRSR moving forward. And lots of outcome measures groups happening. I know there's one with the U.S. government. The Alliance also has one, the International Alliance, bringing together a lot of those people to try to make better outcome measures for ALS. Okay. Number 45, how many steps are in the multi-step model proposed by Amar Al-Chalabi? How many does a pathogenic SOD1 variant account for? What is Amar Al-Chalabi's stage name of his musical act? I'm blanking. I think it's 6 steps. Oh my gosh. And the SD1 variant accounts for 4 of those. Gosh, I hope that's right. Look it up. Maybe I'm being a bad quiz master here. I don't have the answers in front of me. I'm just going off my top of my head. It's either 5 and 3 or 6 and 4. I think it's 6-step method, and it's 4 of those by SOD1, more than any of the other genes. The idea behind that is that, if you have a genetic variant that confers high risk for ALS, it means less amount of other things, environmental impacts or whatever, would need to trigger the disease in this multi-step model. So, for example, somebody who doesn't have anything that's a high genetic risk individual sort of variant in a known ALS gene may be starting at 0 or 1, and until we understand better the more complex genetics, and then what are the different steps that have to go forward in terms of triggering the disease. Whereas in this case, I think C9 was 2 or 3. Gosh, I should have looked this up before doing this, whatever. But I think SOD1 was 4, and it was more than any of the other ones. So it's just a question, whatever. Oh, and the stage name is Blue Sky Effect. Go look up the song Icarus to Your Son on YouTube. I don't know if he has albums for purchase. Buy them, because Amar's awesome, and we should support him. He does all his recording and producing and everything in his home by himself. Yep. He's awesome. Okay. Number 12. Who was the corresponding author on the first C9orf72 molecular biology paper on dipeptide repeats published in Science in 2013? This was a big deal. This was in the Science. It came out. It was dipeptide repeats. It was Dieter Edbauer, German researcher. And this was sort of the first time. So I think once C9orf72 was discovered to be expansions causing ALS in 2011, most of the research that happened was looking at people who were in these families and trying to understand it at the clinical level. But it takes time to be able to make models and to be able to understand what's happening at the lab level and start to sort of look at what's happening. There was a strategy that happened back in 2011, I believe. This was just before I started ALS Canada. I would have loved to be in that room about how we're going to approach this new, very important, most prominent genetic cause of ALS. And I think the first key paper on the biology that came out was this paper by Dieter Edbauer about dipeptide repeats. Again, big tangent we could go on about dipeptide repeats. There is something coming up somewhere in here that we'll also talk about that. Okay, but I got to get moving because I know this has probably already been like an hour. Doesn't matter. Quizmaster can take as long as they want because nobody has to watch. Okay, number 51. What are the names of the Russian scientist who first described neuronal inclusions in ALS and the Japanese-American professor who translated and confirmed her work? It's Tatiana Bonina-Boris, and Asao Hirano, who was a Japanese-American professor who really highlighted this work of looking at these Bonina bodies. I think it was Bud Rowland after that did a lot of this stuff with early aggregates, but this is really that sort of time when pathologically they were discovering these sort of inclusions, aggregates, chunks of protein that seemed abnormal in postmortem tissue of ALS that led to all of the current hypotheses that, this was abnormal biology and this could be something that explains why ALS has, partially the disease that happens and connects to potentially other neurodegenerative diseases. These days, it's always a big question when you have these different aggregates or inclusions in the cells. It's a couple of things. Is it that they're toxic and there's a gain of function? Is it a loss of function because whatever's in there is not able to do its normal function, so it's being sopped up into these chunks? And the other piece is some of these are just like dynamic. We've learned in the past 10 years, a lot of these are dynamic structures, and there's tons of these in the cells that are really complex. And so, are these things that form and break apart? And maybe it was just in that time that they were in that sort of process. So, but Tatiana Bonina-Boris, I'm not sure if I'm pronouncing that right, from the Soviet Union, and Asao Hirano. Okay. 49. OrfAI Therapeutics, Acurastem, and Verge Genomics are all advancing inhibitors of what kinase? It's PIK5 inhibitors. So all of these are going right now. Acurastems, I don't think has reached trial yet. AI Therapeutics had apilimod. Apilimod was interestingly connected to ALS in a Nature Medicine paper about C9orf72 haploinsufficiency by Justin Achita several years ago, where it seemed to rescue some of the the autophagy aspects of the potential normal function of C972. And then he had this big Cell paper, was it Cell? Very recently, where he showed that PIK5 inhibitors really worked in a lot of different models, a lot of IPS models of ALS from different genetic causes and stuff, and through a unique autophagy mechanism, if I remember correctly. Gosh, I got to look at that paper again. Anyways, Verge has VRG 50635. Canada actually has sites for that. Right now, very scientific phase 1 clinical trial. CureStem, I don't know. They've got some proprietary ones. They were looking at apilimod that's connected to Justin Achita, incredible researcher at University of Southern California. And AI Therapeutics ran the apilimod, and Suma Babu from MGH, I think, was presenting a lot of that data last year. I don't know what's happening with apilimod and that one moving forward. I'd have to ask. So, okay, number 59. What nucleopore basket protein was discovered by a Canadian PhD student in the Project Mind data, formulating a large part of her thesis, submitted a couple of years before it was first published and heralded in bioRxiv, and then now in paper and published paper by Luc Dupuis, the awesome French researcher on ALS? And the answer is NUP50. NUP50 was actually something discovered and talked about in a thesis that's online by Jessica Morris, working with Christopher Shaw and Cheryl Gregory Evans in Vancouver. She found it in the Project Mind data and worked with it through her thesis in a completely different and unique way, but it didn't really get a lot of, I don't even think it became a paper. Can't remember if it did. I don't think so. And then when Luc Dupuis found this is obviously really important because now it's a NUP, which nucleocytoplasmic transport is something that's one of the hypotheses is being disrupted and a potential therapeutic target. A lot of work from Jeff Rothstein and others through the Packard and Alyssa Coyne. And so the idea that there was a variant that could cause ALS that was in a NUP or a nucleoporin was a big deal. And Luc Dupuis will always get the credit for that. And Luc is great, and Luc should get credit for a lot of stuff. But interestingly, Jessica Morris discovered this in the Project Mind data, just the public data that she was looking at. It's really cool. Shout out to her. She'll never see this because I don't think she's an ALS anymore, which is, I guess, a shame. Okay, number 26. What is the name of the lactate transporter implicated in a 2012 Jeff Rothstein paper, Nature paper, where oligodendrocytes lose their ability to support neurons in ALS? MCT1, I think. Yes, MCT1. So oligodendrocytes, the forgotten cell type. We don't do a lot with oligodendrocytes, even when we're making models of the disease, like 3D models and multicellular models in the lab, where it's usually microglia, astrocytes, and neurons. The oligodendrocytes really, the myelinating cells, so often connected more to MS. We look at oligodendrocytes, but this paper in 2012 was saying that this nourishing MCT1, if I'm wrong on that and you're all laughing at me, that's fine. MCT1 was not doing its job properly in oligodendrocytes, and that might confer vulnerability to the disease in the motor neurons. So don't know what ended up happening in that. I know that Jeff really likes oligodendrocytes because when someone talks about them in a talk, he's always at the microphone. So there's something still there, but maybe it ended up being less of a exciting pathway than nucleocytoplasmic transport, which became the big drive for Dr. Jeffrey Rothstein at Hopkins. Okay, number 25. What is the primary non-ALS related phenotype associated with the A315T mice under the prion promoter, and what is done to rectify the problem? Bad pun there. It's intestinal dysmotility, and you rectify the program with a jellified diet. So basically, this is one of these ones I talked about before, where the mouse is massively expressing the protein TDP43, and it's just a wild type protein. It doesn't even need to have the mutation or the variant, and it can cause this sort of motor abnormalities. But what they found first is that the mice were dying young, and it was dying because they were, the intestines weren't properly moving stuff through, and that was bad for the mice. And I think it was nitric oxide neurons were degenerating that are affecting the ability for this the actual motility of the intestines. And then it was found that when you don't give them a solid diet, give them a jelly diet, this actually could allow them to survive past the dysmotility and end up getting, phenotypes that were sort of mimicking ALS, which allowed it to become an actual model. Interestingly enough, there's some work being done by Minden Nguyen in Calgary, who he says that in his hands, you can give them the non-jellified diet and they do not have the intestinal dysmotility. So there's lots to learn there. He's looking at microbiome. So I don't know. I've got to learn more about how that works with him. But this is definitely, anytime someone writes a grant and they're using the A315T mice, I know in Canada we have Jean-Pierre Julien has one. There's others. People always say you got to make sure that you're using this jellified diet. That's what the peer reviewers will always say. It's a big deal. And if you're not saying that you know how to do it, there's always a concern that you might just hit this roadblock where there's like really rapid dying. And I think it's even much more in the males or something than the females. Anyways. Okay, number 55. What is the name of the urinary biomarker that rose to prominence for a time after its discovery by Mary Louise Rogers in Australia? That's P75ECD, or extracellular domain. This was work done with the Miami group with Joanne Wu and Michael Benatar. I think it was meant to be like a prognostic, a pharmacodynamic, and a disease response biomarker. I'm not sure what the current status is. I know that I think it was tough because I think Mary Louise in Flinders was needing to be the one that did the actual assay. I'm not sure if now that's changed. I think it was supposed to be in the Healy platform. It probably is, and there's people who will know that it is. I'm not sure how much it's getting used moving forward. But definitely is a big deal, and it's still something that I think is involved in some aspects, but it'd be a good question for me to find out more on. I wrote the question. I should probably know more about it. I forget. Okay, what is the name, number 32, what is the name of the large cholinergic synapses that modulate motor neuron activity and have been demonstrated to have altered activity in ALS by research in Nova Scotia? The answer is C-boutons, and it's Turgai Akai, researcher at Dalhousie University. These are these large cholinergic sort of neuron-like things or inputs that are kind of like volume knobs for the motor neurons. And so I think the idea had been that maybe when there was degeneration of the neurons, that they were almost being turned up in a way that might compensate or lead to the compensation of the neurons to sort of like take a degenerated neuromuscular junction, sort of reinnervate that to compensate so that people, even though they were losing neurons, they were still able to move. And that's sort of the distance between losing neurons and actually having symptoms where the muscles start to become paralyzed or weak. So he's done a lot of work with this. And I believe, in fact, it turns out that in the ALS models, I think he's used a SOD1 model, G93A, that the C-bouton increasing was actually not good. It wasn't a good thing. Like blocking it was better. And then I think blocking it with exercise was even better. And so he's trying to understand what was the exercise creating. And I think it was like serotonin or something. And trying to find a mechanism between C-bouton blocking and some pharmacological mechanism you could do to actually treat ALS, because obviously you're not going to prescribe like aggressive exercise. But in the mice, it was like if they swam and you blocked the C-boutons, they were actually really did better. It's really cool. It's quite complicated, actually, I think, to try to figure out. Maybe not if you read it, you'll go, it's not complicated all day. You just need to read it again. Okay. Number 36. Oh my gosh, there's still a lot of questions. Number 36, the MNDA Association. The MNDA, which is the MND Association, has had a famous patron since 2008 who's a doctor, but strangely not a real one. Who is it? Benedict Cumberbatch, who is Doctor Strange, but he's not a real doctor because Doctor Strange doesn't actually exist. He's a really big proponent for MND. MND-ALS, and there are other patrons that are well known. Eddie Redmayne, obviously, he portrayed Stephen Hawking in and won an Oscar for it. Lily James is also a patron for MND, which is great. There's another one coming up, I think, in another question, so I won't give that away. But Benedict Cumberbatch, Sherlock Holmes, and several other acting roles in movies. Okay, what is the number 13? What is the primary complication in targeting antisense strand DPRs in clinical trial compared to compared to the previously tested sense strand DPRs? These were clearly things I wrote quickly for me to be able to read off to a group. So with compared is just poor. Anyways, it's they're too low concentration. It's a weird pub answer, but the answer is that they're too low in concentration. So poly PR, poly PA are really hard to detect with an assay. Go talk to someone like Tanya Gendron at the Mayo in Jacksonville, who did the assay for the Biogen trial, Biogen Ionis's sense strand DPRs, and it's just they're super low, like it's almost undetectable. So I think they are going to knock down the anti-sense strands. I'm pretty sure that's the next step that's going to happen. I know a lot of people are talking about this. Ionis will probably do this. I think I don't know who's doing it, but I know Jeff Rothstein talks a lot about this being the next thing. It's going to happen. And I think they'll just have to, if they can't get the assay to work, I think it's pretty much like they'll just have to surmise that these are going down or find another way to show target engagement. But, poly PA, poly PR do seem to be quite toxic. I think that's the idea. And then the other possibilities, do you have to knock down both DPRs, or is it too late, or all these other things? But, we've done the sense strand DPRs. That was the wave in the Biogen trials that both were negative and were stopped after phase 1. So now antisense is the way to go. Just harder, because the great thing about those ones, which isn't published yet, but it should be published soon, I think, is that the sense strand DPRs were actually shown to be robustly decreased in those clinical trials with those ASOs, which is nice, because then you're testing the science and really making a decision based on knowing that the sense strand DPRs were actually decreased in any sense strand, probably like RNA biology as well. Okay. Number 9, which inflammatory pathway was first connected to ALS and C972 models by Bob Balow and has subsequently been found to be disrupted in other genetic models of TDP43 and SOD1? The answer is C-gas-STING. C-gas-STING. So this is the mitochondrial related leaking of DNA causes this inflammatory mechanism. And it was interesting because it was the Bob Balow showed it in C9 knockout mice, which said that, C9 may be preventing this sort of immune exacerbation. And then Seth Masters showed it in TDP43 mice. I think it was the overexpressing mice, the prion protein overexpressing, not prion protein, but the prion promoter overexpressing gain of function mice. And then SOD1, I think, was not published yet, but it was some Canadians who were able to show this as well. Honglin Lau and Neil Cashman. And yeah, so the idea was that STING inhibitors or C-gas STING inhibitor pathway inhibitors might be something therapeutically in ALS. I've heard that there's a potential that this has been difficult to get forward to clinical trial, maybe. I think they're being advanced STING inhibitors for cancer. So I don't know what's happening exactly with that, but that's something that at least was a really big deal. A couple of years ago, it was published in pretty high impact papers. I think it was like Nature and then Cell for Seth Masters in Australia with the TDP43 mice. So C-gas STING, stay tuned, might still have some prominence in ALS as a target. Okay, number 4, who is the current primary ALS clinician expert key opinion leader in Belgium? The answer is Philip Van Dam. And there's not much more to say there other than he's awesome. And if you ever get a chance to learn from Philip Van Dam, you should, because he knows a lot. And he's a really big part of TriCals and NCals. And yeah, Belgium actually has a lot of great stuff going on, not only at the clinic with trials with Professor Van Dam, but also Ludo van den Bosch there in Leuven, Sandrine de Cruz, also in Leuven. Previously, Wim Robrecht, lots of great stuff. Belgium, awesome for ALS. Okay, number 53, what class of compounds were tested in some of the first published ALS clinical trials in the 1970s and are also currently being tested in the Lighthouse Phase 3 clinical trial as antivirals? The ones back in the 70s, I think, was like Tiloro, I don't know, something along those lines. It was an interferon inhibitor, so different type of antiviral, but there was the idea that viruses might cause ALS, like, are in old review articles in the 1970s. And so this idea came back with the idea that there were HERV-Ks, reactivated retroviruses in ALS, Avi Nath. We got Renee Duville in Canada, in Winnipeg. Some work done by Amar Al-Chalabi alongside, I want to remember There's a really incredible scientist in the UK. Oh no. And she looked at reverse transcriptase and anyways, there's a lot of evidence to some extent that there could be reactivated retrovirus, and whether that's toxic or not, whether that's conferring anything to do with the disease is the question in certain individuals with ALS. And then, this led to the idea that you would put Triumec, which is a triple therapy, HIV therapy, into ALS. And there's some anecdotal individuals who have been on this type of stuff for years, and so there's always this desire to kind of look into it more. And the Lighthouse trial had an interesting phase 2 and has led to this big, long phase 3 clinical trial that's happening between Australia, I think, with Dominic Rowe and Utrecht in Tricals and Australia. That we look forward to seeing the result, because that's a big deal to be able to see that and kind of at least get a good feel for whether or not there's any value in looking at antiretrovirals and ALS. So glad they're putting that in the big, robust trial that should get us an answer. Number 2, what Canadian primarily cancer researcher published on FUS autoregulation 2013 leading to a flurry of media in Manitoba? The answer is Jeff Hicks. It was an interesting piece. It was in PLOS Genetics. And it was able to show, like TDP43, that FUS could autoregulate itself. So, if it was clumped up in these clumps in the cytoplasm, then you would have excessive production of FUS, which could lead to sort of an overexpressed toxicity. So it was an interesting piece that, he was able to find this, but I think it came off in the media and like on TV news that this was like the next revolution of therapeutic for ALS. Interesting quick story. I actually had to go to Winnipeg a week later and give a talk, and Dr. Hicks was there. He's super nice about it, but I had to make a whole bunch of slides to sort of temper it, which wasn't fun, because I think the audience were really excited and buzzing about this. And there was even a politician who said, isn't it great that the cure might have come from right here in Winnipeg, right before I went up. So one of those fun experiences. Yeah, so that's just to say we need to keep doing good communication out to the community to make sure we balance out, the important and exciting discoveries with what they necessarily mean for us and from a therapeutic sense, and timelines and what does actually mean in terms of its clinical relevance. Okay, number 42. A Japanese group led by Hideyuki Okano published in Nature Medicine 2018 on, I should say, when I start reading these, I hope I can answer them, because I wrote them at the time, but then I worry that I'm going to do this and I can't even actually answer them. A Japanese group led by Hideyuki Okano published in Nature Medicine in 2018 on iPSC models for drug screening derived from individuals with sporadic ALS. It was a big deal as a Nature Medicine paper, saying that, they were looking at sporadic, taking blood or skin, I don't remember which, to make human neurons from people with sporadic ALS to be able to look at sporadic disease in a larger group, and doing drug screening. What dopamine agonist was presented as the lead compound from the work to move to clinical trials, currently in human studies. I think, I'm not sure if that's public, so if people don't see that, just ignore that. Currently in human studies, the answer is ropinirole. I don't know if the other part's true, so just ignore it. Ropal's trial. Yep, that's about all to say. Ropinirole was what kind of came out of that. It's in Japan, and it went into its clinical trial, and I don't know what the fully results were. I think that was promising enough for them to move to the next step, but I don't know whether it's happening or what's happening with that. I have to look it up. But it's like I think the only thing with ropinirole is that it's available for something else, like for Parkinson's, but I think there's some it's careful safety and tolerability, so not something anyone should go do, because obviously this comes from cells, but now in clinical trial. Okay. Who started his career? Number 29. Who started his career at ALS TDI close to 25 years ago, was best friends with the president of TDI at the time. Spat poorly written things. Sean Scott, who sadly died of ALS and continues to now serve as CSO and CEO today. Bonus if you can name the target that TDI advanced to clinical trial. Bonus if you can name the monoclonal antibody drug name moved forward through phase 2 by Eledon Pharmaceuticals. Fernando Vieira. Look him up. Awesome. Super great guy, and just like really super knowledgeable and really objective about the way he approaches ALS. Just a gem of a person to be working on the disease through TDI all these years. They used to do all those mouse screening of all the things that would come out of the literature and seeing if they could verify it. And after all that work, they were able to move forward the CD40 ligand inhibitor antibody, monoclonal antibody to block CD40 ligand as a sort of an anti-immune inflammatory piece, when Steve Perrin was there as the CEO, through to clinical trial, which is a big deal because if they're going to do all this screening and they found all these things to be negative and actually got to one that they felt this is validated, no matter what we do, this looks like it's a positive thing, they get to clinical trial. That's a big deal, and it's a lot of weight to put behind that after years of not having something that they did advance because they weren't sure about other things. And then this became tegoprubart, 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 I think is how you say it, through Eledon. So it was really cool because this one had like great target engagement. It reduced a lot of different things. Actually showed a number of immune and inflammatory markers that were reduced. And then I think I don't know what's happened with it. It's stuck. It like didn't move to the next stage, which is for whatever reason, hopefully we find out eventually, or whether it does, because I thought this was going to be one of those great kicks of the can for the dampening of the immune inflammatory response to say like, look, if this doesn't work, we really need to stop doing generic sort of dampening of this. And also given the great biological, like sort of basic research that was behind it, that would tell us a lot. So I was kind of disappointed this didn't get through because I was really interested to see what would happen. So hopefully it does. Hopefully we can revive tegoprubart and learn more about it and what it can tell us about immune inflammatory response in humans as a therapeutic target for ALS. Yep. So there's that. I wish I could remember the name of the company before it became Eledon, or before it got purchased by Eledon. Shoot, I should know that and what it was called. I used to know a lot. Anyways, okay. Number 22. What Canadian science legends pursued glia in ALS and other neurodegenerative diseases years in advance of Don Cleveland's seminal science papers? Bonus if you can name the sport that one of them represented in Canada in the 1948 Olympics. Bonus number 2 if you can name the other ALS neurologists who competed for Canada in the Olympics in Vancouver 2010. All right, that's a lot. First question and point is Patrick and Edith McGeer. So Don Cleveland had these really big science papers. Albrecht Clement with astrocytes and Séverine Boilet with microglia in like 2004, 2006, where they made these chimeric mice to be able to show what's the contribution of astrocytes to like the triggering of disease and microglia to the progression of disease. It was super eloquent work. It was a really cool way of showing that glia it was a non-cell autonomous disease. It wasn't just the motor neurons, but the glia were playing an active role. These were seminal papers. These were big papers that like I think we harken back to now in terms of like where we are in the disease and everything. Like, it's a big deal. I mean, harken's a word. Who says that? Anyways. But if you look upstream of that in the literature, in terms of saying that ALS and these neurodegenerative diseases had a contribution from the glial cells, actually Canadians, Patrick and Edith McGeer, really did a lot of really seminal work on this in the background before that. So definitely worth looking them up. And Patrick McGeer played basketball in the 1948 Olympics, representing Canada. He was a tall man. Yep. So that's that. Some Canadian neurological legends there. Bonus number 2. You're probably not going to know this if you're not from Canada, or you probably don't know if you are from Canada. We had a cross-country skier in Vancouver 2010 who's currently an awesome neurologist in Calgary working on ALS, and that's Gord Jewett. Gord actually works on wearables in ALS. He was a recipient of a Mitsubishi Tanabe Pharma Canada Clinical Research Fellowship, and which was delivered through an ALS Canada program that they supported with the funds. And he's just incredible. He's really been doing a lot since he started just a couple of years ago, and also an Olympian. So that's the answer to that. Number 38. Individuals who are homozygous for what allele at SNP RS 126089 08932 of UNC13A are both at risk of shorter survival with ALS and currently enrolling in the Prelude clinical trial using this genotype as a predictive marker for potential lithium carbonate, David. Oh my gosh, not chloride. Preform David was not smart. Lithium carbonate efficacy. Okay. The answer is the CC allele. So UNC13A, many of you will know. Oh my gosh, there's a lot we can unpack here. Looking at the time, I don't know what the time is, how long I've been doing this. We're getting closer though, the end. Okay, so UNC13A was shown that if you have a CC SNP, so a little change in a piece of the DNA, or you have a CA or you have an AA, it all could actually change the progression rate of the disease, and that people were faster progressing when they had the CC allele. And so that was always a question as to why that was, but it was very interesting. Michael Van Ass in Utrecht, really cool stuff. He was looking at clinical trials that were negative and trying to look at the genetics of the individuals who were involved to see if there were responders who had specific genetic makeup. Really cool. So there were a couple lithium carbonate clinical trials. I know we're getting off on a big tangent here. Italy showed that lithium carbonate was actually protective in ALS in like a phase 2, phase 3 study. That got everyone very excited and drove Niels and Cals, actually, the Canadian ALS Research Network, to collaborate on a North American lithium carbonate clinical trial that was stopped for futility because it really wasn't doing anything. And so procuring the data from those various clinical trials that happen on lithium carbonate, Michael Van Ness found that individuals who had the CC SNP allele for UNC13A actually seemed to do better than those who had the CA or AA. And so were they responders? Was this actually something that connected to lithium chloride potentially having efficacy? And maybe there was difference in the Italian populations that were involved in that trial versus those in the North America. All of this to say maybe it's a responder group. And that's where the Prelude clinical trial comes in. It's super cool with M&D Association and TriCals. It's like their magnet platform, whatever. Prelude's sort of the first big one on that. And they're recruiting only people with the CC, I believe, to see if, placebo versus lithium carbonate, if there was a slowing of disease, which would be super cool because it would be the first time we have a predictive genetic biomarker for a potential therapeutic in ALS. Now, we all know we could go into a huge bit about UNC13A. It's now a therapeutic target, restoring it, potential synaptic activity. The work of Pietro Fratta and Aaron Gittler have shown that it's actually that when TDP43 is no longer in the nucleus or when it's reduced in the nucleus, you get cryptic exon inclusion, you get UNC13A loss. So and that the cool part here is that the alleles, the ones that confer the shorter survival, tend to have an effect on the TDP43 binding, according to those papers. So even when you have as much TDP43, but you have the different CC versus AA, the TDP43 can repress more of the cryptic exon when you're at AA than it can with CC, so that exacerbates the disease and kind of explains the potential for the shorter survival. So the idea being now that you can restore UNC13A as a potential therapeutic target, something that some of the companies are working on. Actually is a big deal right now this week because Lilly just bought this from Curalis as an ASO to restore UNC13A. I think Curastem's also doing this. So we'll see in the days ahead, but lots we can unpack there as well. Number 11, name the M&D investigator at Macquarie University in Australia who's been able to use ancestral pedigrees to connect people with seemingly sporadic ALS as actually familial in nature. And the answer is Kelly Williams. She has super cool things. If you want to go on, I'm sure she must have something on YouTube I haven't seen, but I've seen her, give live talks, where in Australia she's been able to actually look at individuals that would be basically classified as sporadic ALS, but it turns out that they actually are connected to other individuals with ALS if you look in a deeper pedigree, like I guess what you call super pedigree. And this is actually really cool too, because what it's going to tell us as we look at some of these is maybe that, for certain people with ALS that have a genetic background or a genetic susceptibility to ALS, it's more complex, and it's not just a single gene passed on from a parent to child with a variant, but in fact, some of those complexities might be told by looking at bigger pedigrees of people to see what is that key cut that makes that one person susceptible through multiple genetic avenues as opposed to just one particular gene. It's like raining outside. It's very dark all of a sudden, and I'm not going to get up and go turn on a light, but it's here in Scaryville. in my basement. Okay. Number 16. Which member of the British royal family is a patron for the MND Association? Scotland. Oh my gosh, I made another mistake. MND Scotland, I believe. Yes. And that answer is Princess Anne. She was actually in Edinburgh. No, she was in Glasgow at the ALS MND symposium several years ago, hosted by MND Scotland. Super cool. She was there and gave Dr. Angela Genge the Forbes Norris Award. Presented it to her, I believe, which is also super cool. Canadian. Oh, okay. This is when you can't record something because things pop up on your screen. Okay. What ALS expert also founded and leads Team Canada Healing Hands, a nonprofit that provides rehabilitative education, training, and care in areas of need like Haiti? The answer is Colleen O'Connell. In Fredericton, Colleen is incredible. She does work in Asia. She does work in Haiti for rehabilitation. Pretty awesome stuff. She's also a physiatrist that leads the Stan Cassidy Center for Rehabilitation ALS Clinic, brings clinical trials to New Brunswick. She's part of an amazing initiative about how to break bad news for diagnosis with Angela Genge and Kathy Cummings and Melinda Kavanaugh. She's just a gem of an individual and does incredible work. And so Colleen O'Connell needs a little more recognition in our field. Okay, number 52. What is the name of, she's also doing this thing with the VR. It's really cool for quality of life, better breathing for people with ALS. Okay, getting there, almost done. What is the name of Ionis's FUS ASO, previously known as J-Suffusin? It's Ulefnersen. The name J-Suffusin obviously coming from J.C. Hermstad. Amazing story. You don't want to have me retell it here, but really hits the heart. And obviously combining that with Anna Kempfer and her story in Germany right now, who is seemingly rebounding with FUS, very aggressive FUS variant after having Ulefnersen, and there's several other people. We're really excited to be able to see what will happen with this one. We don't know yet, obviously, what will happen with Ulefnersen, but clinicians seem to be seeing things with some of their patients that they've never seen before with FUS, especially those rapid progressing individuals who, get a very young onset and progress very quickly. But we will see. It's one of the big hopes we have. Also, neurofilament decrease with FUS ASO from Neil Schneider's talks and work. So that's a good second validation of the value of neuropreservation with NFL decrease beyond tofersen. Okay, number 6. Which ALS researcher propagated the work from Chicago to Montreal to Toronto to Vancouver? It's a stupid little pun in there. The answer is Neil Cashman, another Canadian-centric one. Neil is one of the biggest proponents and drivers of the idea that SOD1 and others are sort of propagating in a prion-like mechanism, the misfolding of proteins. So he's done a lot of work on that. He's now chief medical officer, I believe. Is he the CEO or the chief medical officer of oh my gosh, they're developing RAC1 therapy and several other antibodies. And he's gonna hate me that I am blanking on the name of the company. But shoot, something to do with antibodies, I think the name. I should know this. I know this very well. Anyways, Neil Cashman's the answer. And he moved around a lot and led the clinic at the Montreal Neurological Institute for a time, the ALS Clinic, then at Sunnybrook in Toronto, and then for a long time in GF Strong in Vancouver. Done a lot of things over a lot of years. So big props to Neil Cashman. Okay. It's going to come to me at some point. I'm going to hit my head over what the name of the company is, because it'd be good to be able to mention it to people for him. Anyways, about 10 years ago, Joe Beckman stunned audiences by presenting an aggressive ALS mouse model that had full recovery and nearly normal lifespan when treated with a compound that ultimately failed to show benefit in clinical trials led by collaborative medicinal development. The answer to the question is copper ATSM. It was Belgium ALS-MND, I don't know if he presented it before then, but he showed this mouse where he had a mutation in SOD1 and then also in CCS, which is what loads copper onto SOD1. And then when treated, those mice got a rapid motor neuron degeneration. And then when he treated them with copper ATSM, they just like didn't die. It was crazy. It was not a super validated ALS mouse model, but it was still the dichotomy between the treated and untreated mice was something like we'd never seen before. And the cool thing was that TDI, who I talked about before, actually were able to replicate that in G93A mice. There was a benefit of copper ATSM. So when this went to the Australians, group on collaborative medicinal development to put this into clinical trials, was actually pretty exciting. Actually, I always felt it should be done with an extra arm of SOD1 just because the copper piece. But now we have other therapies for that anyway. But anyways, this was the one that in phase 1, it got all hyped up. It was 70% slowing of disease, but it was open label. It was very small, very underpowered. There's a long story behind that as well. But now it had been into a slightly larger phase 2, and there just has been no word from it. But word on the street is that it was negative and just never really saw the light of day. Whether it will at some point, we don't know. But really, it's just not something that CMD is pursuing anymore with copper ATSM, as our understanding, but had that really exciting data back in Brussels back in, 2015, 2014, something like that. Okay, number 17, almost done. Who has been the PI of the Swedish ALS-MND registry since its inception in 2015? The answer is Caroline Ingra. She's awesome. She's a star. She's going to be big. She is already a big part of TRICALS and NCALS and will be one of the big leaders for the foreseeable future. And does work on early diagnosis, does work on clinical trials, does work on great clinical research and procurement of data for different things, and also leading this registry for many years. She's been super awesome to get to know and work with on the Scientific Advisory Council of the International Alliance of MND, ALS-MND Societies or associations. And if you don't know who she is, you should, because she's awesome. Okay, number 34. What is the name of the hormone that last year's contestant on the Showdown, Vincent Pichot-Martel, coming back to actually be a neurologist in Quebec City from his fellowship at MGH with Merit Sokovich and Clotilde Lagier-Touraine, super cool, in collaboration with Yasna Kriz, has shown as dysregulated due to immune profiles in ALS and may be linked to disease progression? The answer is leptin. Yep. So leptin, I think, was decreased in people with ALS, and there was an increase in AMP kinase, in the adipocytes when they looked at this, and that it was believed that this might actually be a biomarker for ALS, that the decrease in leptin. I think I got that right. I've always been a little confused about that one. It's complicated, that whole system, because the AMP-K, NAD whole bit, some people are trying to boost that, while this one would say that it was actually, I think, bad to have increased AMP-K, and when they inhibited, they saw the leptin levels came back, and this might be a pharmacodynamic biomarker. I can't remember if it was prognostic as well, because I think it had related to metabolism and faster progressing, had lower levels of leptin. I can't remember exactly. Go look at the paper. Again, can't know everything. Yep, number 18. What's old is new again? What target of the late 1990s and early 2000s is currently being pursued by Amylyx with the ASO AMX114? The answer is calpain. I think that's getting closer to the clinic now. Calpain was something that, people looked at and talked about for a number of years, including the lab I worked in for my PhD, did calpastat and calpain mechanisms. Miranda Tradewell, working with my boss, Heather Durham, in motor neurons, and those mechanisms are looked at over a number of years. But calpain, obviously not a great thing, calcium-related cell death mechanisms. So blocking that might be something that could work. We're going to find out from Amylyx, hopefully in the days ahead. Yep. Number 35, the key individual driving both early ASO lab work for ALS and treatment of pseudobulbar affect left clinical duties to work on these things in the lab. Earlier in his career, he shared the clinic with what neurologist who is considered one of the founding individuals of multidisciplinary care? That's a really tough question. So the key individual is Richard Smith, who was working in San Francisco in the clinic as a clinician, as a neurologist. He left the clinic to go advance ASO work with Don Cleveland in the back of the lab, as well as Nudexta, So dextromethorphan and quinidine combination for pseudobulbar affect and potentially for slowing bulbar symptoms. Yeah. And those are huge things, really human impactful things, massive. Shout out to Richard Smith. If you don't know who he is, look him up too. Anyways, the question was around early in his career, he shared a clinic with what neurologist is Forbes Norris. Forbes Norris is considered to be sort of one of the founding individuals of multidisciplinary care, one of the first sort of ALS-focused dedicated researchers. And Forbes and Richard Smith actually used to work together. And Richard was always the belief of the neurologist being the lead kind of person, and Forbes was always the person like Arthur Hudson, like Ted Munsat, that sort of took the ideas from muscular dystrophy in the UK, and I can't remember the gentleman's name there, of multidisciplinary care and the need to have multiple individuals who are really seeing these individuals. A super cool thing of Arthur Hudson in Canada in 1978 or whatever in the London Free Press newspaper, where it's him and his entire team of multidisciplinary care talking about how important, an OT was and a PT and a respiratory therapist and all this, like super, super cool old article that I have on multidisciplinary care. But anyways, the answer is Forbes Norris. Number 47, from what study is Janine Heckman leading efforts to stimulate genetic discovery and networking for a whole continent? Cape Town, South Africa, for all of Africa. She's trying to build ALS Africa Net, or the network of group of clinics in Africa, to build infrastructure, to build clinical research, to get better communications that are applicable to each of the different nuanced cultures and languages in those regions, which is, we in North America and Europe probably can't even conceive, I think, of the complexity of all that at so many levels, but trying to start that and bring it together. Great grant by ALS Association to get that going. Hopefully she'll continue to get the funding to be able to get that going. She's super big leader out of Cape Town, and got together, I think, groups in in Nigeria, and in Kenya, and then another group in South Africa, I think in Johannesburg, and then possibly someone in Tanzania. And then I know there's some work being done in Ethiopia, but those are, so different, all those different regions. But starting to build that kind of a networking to move things forward in Africa, because just like I said with South America earlier, we want to see clinical research. We want to see better clinical infrastructure. We want to someday see clinical trials and treatments available to people all over the world. So that's super cool. She's like a superhero of mine. Okay. Stan Crook is credited with the vision of keeping ASOs alive as a therapeutic modality, but who is responsible for driving Ionis's use of ASOs in the central nervous system? The answer is Frank Bennett. There's an awesome podcast all about Stan Crook and Ionis and driving ASOs forward called Hope, Lies, and Dreams. I think that's what it's called. I listened to it like a year ago. And he says that he didn't have any interest in neurology, in neuroscience. And Frank Bennett said, we need to do this. ASOs could be really good for this. This would be awesome. And he said, fine, go, Frank, do it. And he did. Look at that. Spinraza changing the lives of people with SMA. Tofersen changing the lives of people with SOD1 ALS. Frank Bennett's a legend. And he gave, I think, the best bowling alley talk I've seen at the Packard before, where, I mean, it's just like edge of tears. You're watching. This is one of the first times we were able to see like the kids who were on Spinraza and were just like thriving. That was incredible. So that was very memorable. He came to our research forum one year. We gave him a fishing net, actually. Personalized thing as a gift. Hopefully he's using it. He's a fly fisherman. Awesome, Frank Bennett. Okay, number 43, 2 questions left. We're almost done. What company's motoneurotrophic factor compound, GM604, was tested in a 12-person open-label clinical trial, but sparked petitions of nearly 1 million signatures calling for its approval by the FDA? Genervon. So that's a whole story. GM604 is the name of the thing. You can look up the ALS Untangled bit on GM604. Genervon got everyone hyped up and riled up on the belief that this was changing everything for ALS with a very small number of open label individuals. Run at Columbia with Hiroshi Mitsumoto and Meritsukovich at MGH, and just went around them completely to put out press release after press release to talk about how great it was and really riled everyone up. And it's a really good example of how we have to be super careful on how messaging goes out, because it just isn't fair in these situations. And yeah, lots of background there I won't go into, because we're on the last question now. The last question is, what is the name of the gene with ALS causative variants discovered by Canadian Sally Farhan? Bonus if you can name what organ it affected in its knockout mice. The answer is DNA GC7, and it causes steatosis in liver when it's knocked out. Yeah, so I know people are exploring DNA GC7's connection to ALS in the biology. And that's it. Pretty anticlimactic ending. But I don't know. How'd you score? I think the score is 84. Check it out. Let me know if you ever watch this. That was long. Oh well, it was fun for me. And that's the end. That's what we did. Well, maybe we'll do it again next year and we'll have more questions. Hope you enjoyed it. Okay, bye-bye.